we are going to look at the microscope and how we use the microscope to look at different structures. So first of all, we can see here the basic layout of a microscope. We have the eyepiece lens, the coarse adjustment knob, we have the high and low power objective lenses, we have the stage where you would put your slide, and then we have the light shining through from the bottom through the stage onto the specimen, allowing us to see the cell that we're trying to look at. Now normally what you would do is, when we're using this light microscope, you would have probably looked at an onion slide at school, and all you do is you have your slide, you use a pair of tweezers to get the onion slide, and you put the specimen onto your slide to start off with. Then, because the slide is clear and see-through, we tend to stain the onion skin there, and what we usually use to stain the onion skin is iodine, because it gives it a nice deep brown colour, so we're able to see it through our microscope. Now, to stop the specimen moving, what you would normally do is you would grab your cover slip and you would put your cover slip on top, and you would just gently push out any bubbles so you're able to focus on a clear specimen when you put that onto the stage in your microscope. Although the light microscope is great and it will allow us to see the cells in the onion cell, it is not going to allow us to look at anything smaller. So all the other organelles that we've been looking at, so things like the mitochondria, the plasmid, the chloroplast, so on, cannot be seen with this light microscope. So we've had developments where we are able to see structures within the cell, and that is through using an electron microscope. And in comparison to the light microscope, the electron microscope has a much higher resolution. So you can magnify much higher. It will show a sharper image. So the image is much better compared to the light microscope, and therefore it would allow us to see much smaller detail, so we can start looking within things like cells. So for example, we would be able to see the mitochondria, we would be able to see ribosomes, chloroplasts, etc. So much smaller structures within the actual cells, much higher resolution compared to the light microscope, and a much sharper image. Okay, so now what we're going to do is we're going to have a look at calculations to do with microscopes and magnification. So the first thing that we need to know about is an equation which relates everything. So magnification is the image size divided by the real size of the specimen. Okay, so let's just jump right in with how this could come up. So in terms of an example, put one here, could be something like we've got a specimen and our specimen is 60 micrometers wide. Okay, when we looked at it in our microscope, it was magnified 100 times. And we want to work out what the image size would be. Okay, so the two things that we have is we have the magnification and we have the real size. So if we rearrange the equation, we would have magnification times the real size will give us the image size. Okay, now if we look at this, then we would have the magnification is 100 times the real size, so 60 micrometers, and that's going to give us our image size. And if we work that out on our calculator, we would get 6,000 micrometers as our image size. Okay, now the question might ask you to give your answer in millimeters. Now the key thing about changing micrometers into millimeters is you would need to divide by 1,000, okay, to get it into millimeters. So if we needed to have this answer as millimeters, you would have 6,000 divided by 1,000, and the image size, if we did that, would end up as 6 millimeters. Okay, it is important for you to know your conversions. Now we're going to do some calculations to do with magnification. First thing is, it's important that you know the equation. Magnification is image size divided by actual size. So we've got a photograph of a plant cell. The image size of the photograph shows the plant cell is 15 millimeters across. The actual size of the image is 0.015 millimeters across, and we've got to calculate the magnification and show our working out. So to work this out, we've got to have the image size, which is 15 millimeters, and that is going to be divided by the actual size of the image, which is 0.015 millimeters, and if we work that out, that works out at being 1,000 times magnification. Now if we have a look at another question, and this time it's saying there's a magnification on a photograph which is 500 times. The starch grain is 1 centimeter across in the image. So what we've had to do here now is we've had to rearrange the equation to get actual size as being what we're finding out. So remember, the original equation was magnification, just write that down, was the image size divided by the actual size, and so we've rearranged that to have the actual size as what we are working out, and we're going to use the equation as it is in the box. Therefore, now the actual size, we would have the size of the image, which was 1 centimeter, divided by the magnification, which was 500, and if we worked that out, that would give us the actual size as being 0.002 centimeters. Now quite often we have a question that leads on from that, asking us to do a conversion, which is important, why you need to know your conversions. And now we need to change the centimeters into micrometers. Now to change centimeters into micrometers, what we have to do is we have to multiply by 1,000 to do that conversion. So if we do 0.002 multiplied by 1,000 to get it into micrometers, our answer is 2 micrometers. Hope you found this all useful, and please do subscribe because you'll be able to see the new videos as they come up.